Good afternoon, and thank you for joining us for the Kirby Institute Seminar Series. I'd like to begin by acknowledging the traditional custodians of the lands on which we gather today and pay my respects to their elders past, present and emerging. I extend that respect to Aboriginal and Torres Strait Islander peoples joining us today. So before we begin just some quick housekeeping. So today the format of the seminar will start off with a presentation by Chaturaka, and then this will be followed by a Q&A session at the end. So while the talk is going, you may enter any questions that you have through the talk in the chat forum on the side. So to ask a question, click on the icon with a speech bubble and a question mark in the top right corner to open the Q&A chat screen. When you click on Ask a Question, please remember to write your name, as this will help us reference your question and make answering a little bit easier. You can also upvote any of the questions that you'd like to be answered, and you can also change the order of questions by clicking on the tab on the top right of the Q&A chat, which says Most Recent, and changing it to Most Liked. All right, so just to introduce our speaker today is Chaturaka Rodrigo. He is currently an NHMRC EL investigator, and he's affiliated with both with the VISP program at the Kirby Institute, and he's also senior lecturer in pathology at the School of Medical Sciences at UNSW. So Chaturaka started his career as a medical doctor in Sri Lanka and then specialised in internal medicine, where he gained over 7 years in internal medicine. They gained over 7 years of clinical experience in treating dengue patients. Chaturaka then left Sri Lanka and joined VISP, where he completed a PhD in 2017 on a project that examined the phylogenetic evolution of hepatitis C virus. After completing his PhD, Chaturaka was then offered a combined teaching and research academic position within SOMS. Chaturaka now has established his own research agenda within VISP at the Kirby, and it has a focus on infectious diseases, and this includes hepatitis C, malaria, dengue and leptospirosis. But he still has a particular interest in evolutionary biology of RNA viruses. So as part of this research program, Chaturaka has established a dengue cohort in Sri Lanka, and it is this project that he will be talking about today. So over to you, Chaturaka.
Thank you, Ravina, for that kind introduction. So today I'll be talking about this prospective cohort study that we are doing in Sri Lanka regarding dengue. But before that, since most of you may be unfamiliar with this infection, so I'd like to spend a few moments talking about what dengue is and what is the clinical impact and what is the research question that we are trying to answer here. And then what I'll do is I'll go through a few projects that we are doing in collaboration with our Sri Lankan counterparts, and that will give you a nice idea of where we are heading. So first of all, regarding the epidemiology, so most of you may know that dengue is a mosquito-borne fly virus, but most of you may not know the extent of the disease burden of dengue. For example, there is an estimated 390 million cases of dengue per year. Now the estimates vary depending on different studies, but this is a figure somewhere in the ballpark. Now I say estimated because most of the dengue cases are clinical diagnosis, because in most of these countries where dengue epidemics happen, the facilities for confirmation of diagnosis don't exist, or they're too expensive. So most of these diagnoses remain as clinical diagnosis only. 3.9 billion people are at risk of infection, and acute disease transmission is currently happening in 129 countries, including Australia. So I'll go to in the next slide, I'll explain a bit about that. And there's an alarming increase in the case load. For example, there were only 8.3 million cases reported worldwide in 1990, and there has been a 7-fold increase by 2013. Maybe it's more cases, or maybe it's better reporting, but anyway, we see an increase in the number of cases. A bit about dengue in Australia. So Australia is one of those 129 countries where dengue transmission is endemic. And you see in shaded in pink is the distribution of the dengue vector mosquito in Australia, Aedes aegypti. And you can see it's in the northern Queensland and in the Torres Strait Islands that you get this endemic infection being endemic. Other states also do report dengue cases, but they are mostly imported cases. For example, if you go holidaying in Bali, you might come back home with dengue. Now, dengue is a notifiable disease in Australia. So I think the surveillance started in 1991, and there were only 17 cases reported that year. But the highest number of dengue cases in Australia were reported in 2016, where it exceeded 2,000 cases. And in 2019 also, we had 1,465 cases. It may be a bit less in 2020 because no one is traveling, but nevertheless, it's an emerging problem in Australia also. Now, those who are familiar with hepatitis C virus may immediately recognize the similarity of the genomic structure of dengue with hepatitis C. But when we talk of dengue, and even in phylogenetics, we basically talk of, in terms of serotypes, not genotypes. There are dengue genotypes, but they are categorized within serotypes. And the genetic differences between different genotypes within a serotype is not that great. So, but there are significant genetic differences between serotypes. So, therefore, I mean, most of the dengue phylogenetics, strangely, we do not refer to genotypes, we just go by serotypes. Now, a bit about the vector. There are two vectors for dengue. There's Aedes aegypti and Aedes albopictus. The common name, I think, is the Asian tiger mosquito, and you can see why. And it's a disease of urbanization. So the dengue mosquito actually breeds in clean water. So wherever you get a puddle of water accumulating, and in the rainy season, and if the disease is endemic, you have a high chance of getting making a dengue breeding site. And the more people there are, and the more garbage they throw out. So receptacles use tires, tins, cans, and all these things serve as breeding grounds for these dengue mosquitoes. Now, having said that, dengue is not only a disease of urbanization. You still get dengue in rural areas also, because these water puddles can even form within water collections in between broad leaves of a tree, between foliage. So then, of course, even that amount, is good enough for a dengue mosquito to go and lay the eggs and for the breeding cycle to continue. So therefore, it's very difficult to get rid of the vector. So most countries combat dengue by improved case surveillance and improving their treatment strategies. Now, the clinical syndrome of dengue, I'm going to spend some time explaining the clinical syndrome of dengue because that is quite important for our research question that we are going to see in the later slides. So these two figures are from two WHO classifications. On the left, you have the 1997 WHO classification, and then here we have 2009 WHO classification. Both these classifications are still used by clinicians when it comes to dengue. So they are not mutually. So the old guideline simply didn't replace the new guideline simply didn't replace the old guideline. And I still like this the description here in the old guideline because it gives a more detailed overview of the spectrum of diseases that we see in dengue. So the virus infection could be asymptomatic. We don't know how big this group is because they are not symptomatic. They don't come to the hospital, but we imagine that to be fairly big because there has been serological studies done in healthy volunteers and there has been seropositive care. The seropositivity is quite high in endemic countries. And then we have the symptomatic cases of dengue, and there are three main groups that we can differentiate. First, you may have undifferentiated fever. That is, the fever is not different to a flu or any other tropical fever that you may get, and there are no telltale signs that it's dengue. Or you may get a differentiated fever, which has some features to suggest that you have dengue. This could be like a drop in platelet count, or myalgia, arthralgia, retroorbital pain, and headaches, and maybe the duration of fever. And you may also have a bit of unusual hemorrhage, so which is quite rare with other kind of tropical fever. So then, of course, that is a bit of a differentiated fever, but still this is not a severe illness. Most people will recover uneventfully, so that's a differentiated dengue fever. And then comes the third category, which we call the dengue hemorrhagic fever. Now that is a bit of a misnomer because dengue hemorrhage in dengue, frank hemorrhage, is quite rare. It's uncommon, let's say. What actually happens here is something called plasma leakage. Now, around day 4 or day 5 of fever in dengue, there is this phase called critical phase. Not every patient gets it. About only 20 to 30% of all dengue patients enter this critical phase, which is the hallmark there is that there's an increase in the permeability of your capillaries. So the fluid component of your blood, the plasma, leaks out into the external space, which is the interstitium or the third space. And as a result, the cellular component as a proportion inside your vascular tree increases. And I'll go to that I'll discuss that in detail in the next slide. But anyway, that reduces your effective circulating blood volume. And if this is detected in the correct time, and if you take corrective measures to counter that change in fluid dynamics, then of course you may recover from this stage uneventfully. But if that is not detected in time, and if you continue to leak, then of course the blood, the perfusion to external your critical organs like the kidney may be threatened, and then of course organ dysfunction might happen, and there may be irreversible shock due to hypovolemia, intravascular hypovolemia. And this stage we call dengue shock syndrome. Now it's very rare to get dengue shock syndrome, and we'll come to that in the next slide. But if that happens, that is what usually kills in dengue. Now, so that was a very comprehensive overview of the clinical spectrum. Now, but there was a lot of terminology in this classification. So WHO came up with a more simplified version of classifying things in 2009, and then it was just dengue and severe dengue. Having dengue hemorrhagic fever doesn't necessarily mean that you are going to have a bad outcome. That is, having plasma leakage doesn't mean that you are going to have a bad outcome. It's only if you don't detect it at the correct time, and if you don't take corrective measures, that it ends up being a bad outcome. So a lot of people who have dengue hemorrhagic fever is still included in this dengue category. So just having plasma leakage doesn't mean you have severe dengue. So to have severe dengue, you need to have either severe plasma leakage, severe hemorrhage, and severe organ impairment. And all these things are usually, this number 2 and number 3 are probably consequence of not detecting plasma leakage at the correct time. So in other words, not all patients with plasma leakage will have severe dengue, but almost all patients with severe dengue would have had plasma leakage. So plasma leakage becomes like the least common denominator for having severe dengue. Now this diagram here describes all the things that you need to know about dengue. First of all, it's an acute illness, so it doesn't last more than 10 days. And day 1 of illness means usually day 1 of fever. And you can see the temperature rises usually during the first 3 days of fever, and then it drops actually. So if the temperature drops too early in a dengue patient, that doesn't mean the patient is getting better. In fact, the patient may be getting worse. And that's something that physicians who treat dengue are quite familiar with. And there are 3 phases in the illness. We call it febrile phase, critical phase, and the recovery phase here. Now, critical phase is the phase where plasma leakage happens. And not all patients have a critical phase in their illness. Only 20 to 30% of patients, as I told you, will have plasma leakage, and therefore this phase. Others will just move from febrile phase to a recovery phase. Now, if you happen to have this, then you're at risk of severe dengue. And because of plasma leakage, your hematocrit. Hematocrit is the proportion of the cellular component of your blood. So if your fluid level, the plasma, leaks out into the third space, then of course your cellular volume as a proportion will increase, not because your cells increase, because there's an effectively low blood volume now because plasma is leaking out. And therefore your hematocrit, that is the proportion of cellular component in your blood, will start increasing as the plasma leakage progresses. And this is what we actually use in the clinical setting to identify when the plasma leakage starts in this patient. So, And in plasma leakage, we are not talking in terms of days, we are talking in terms of hours, because plasma leakage usually lasts for only 48 to 72 hours. And anything bad that can happen within this time window. So you need to detect this quite early and at the correct time. And hematocrit, unfortunately, is the only measure that we can reliably detect plasma leakage in the current setting. And we usually say a rise in the hematocrit above 20% of baseline is good enough to make a diagnosis of plasma leakage. Few other interesting things. You see the viremia increases in the first 3 days, and as the critical phase comes, then it disappears, and then the immunological response kicks off. And so this critical phase, if it happens, may be driven by something that happens here, or may be driven by the host immunity. So there may be both components there which are responsible. Now, what happens to patients with dengue? Most people do recover. So it's and actually the case fatality rate, that is the number of people dying of dengue, is less than 1%. So if 1,000 people get dengue, less than 10 will die of dengue. So it's it has a reasonably, it's not a disease that kills like it's like Ebola or something. It's not. To say that it's dengue hemorrhagic fever is actually a bit of a misnomer. There are no chronic stages of disease, but there's this thing called original antigen SIN, which is a real phenomenon. Because if you have infection with one dengue serotype, and if you get a dengue again with a different serotype, the previous infection doesn't give immunity. In fact, it forms non-neutralizing antibodies, which may trigger a dysregulated immune response in the second infection, and you may end up with more severe dengue the second time. There may be a phenomenon of post-viral fatigue, and if I have time, I'll go into that. But anyway, as you can see, there's no chronic stage of illness, short-lasting illness, case fatality rate less than 1%. So why is this a huge problem? Now, dengue is a huge problem, by the way, and the problem is in the numbers. Now, I'll show you a case study in the next slide. And you saw the extremely high number of caseload per year in the previous slide, 390 million cases. And to put that in context, I think the total number of coronavirus cases reported so far is less than 30 million. And dengue annually, the estimated caseload is 390 million. So 1% out of that is still a huge number. So the problem is actually in the numbers. And these cases are not spread out evenly throughout the year. And they come in unpredictable epidemics. So that whole caseload of an year may happen within the space of a few weeks. And that leads to mass admissions to hospitals, because dengue, whenever you have a dengue epidemic in endemic countries, that is also associated with media hype a lot, and people are scared, and the minute they know they have dengue, they come in masses to get admitted. So, and that overwhelms the healthcare resources, and that creates very dangerous situations for both dengue patients and non-dengue patients. And that, of course, creates very dangerous situations where people die unnecessarily. One more thing that I forgot to mention in the critical phase here, if I can go back in this slide, this critical phase only lasts for 48 hours. What happens after that is all that fluid which leaked out comes back after these 48 hours. So if you give, so the treatment during critical phase will be like if you are having too much loss of fluid into the third space, sometimes you may need to give fluid externally, fluid, intravenous fluid sometimes. But if you give too much fluid, and when all that leaked fluid comes back after 48 to 72 hours, your circulatory system might get overloaded, in fact, of too much fluid. And then, of course, that fluid might leak into the lungs, resulting in pulmonary edema, and that might kill the patient also. So they say half the deaths in dengue is due to dengue, and the other half is due to medical mismanagement. So that, of course, you need to be careful. And all those things you have to put in context of these mass admissions that happen. So essentially, though less than 1% of dengue patients die, we don't know which 1%. All the patients need to be admitted. Doesn't necessarily need to be admitted, but that's the usual thing that happens. And that creates very dangerous situations. And here's a case study from Sri Lanka, what happened in 2017. And on the x-axis, we have weeks of the year from 1 to 52 representing the whole year. And in the y-axis, we have the case incidence rate. And the dotted line here is the average of dengue cases between 2012 and 2016. And the broken line on top of that is one standard deviation above that level. And in red is the case incidence, the actual case incidence that we observed in 2017, which was greater than 2 or 3 standard deviation above normal throughout the year. And that resulted in 186,000 cases, reported cases, and about 440 deaths. And all these deaths would have been preventable deaths. So we published this study here in Emerging Infectious Diseases. So the citation is given there, and you can have a read. So this brings us to the clinical question that we are trying to answer with our work, is that are there early risk predictors for plasma leakage? So the idea there being that I told you that not all patients with plasma leakage have severe dengue, but all patients with severe dengue would have had plasma leakage. So if we can at least identify what are the early risk predictors for plasma leakage, then we can at least keep those people in the hospital and send the rest home, hopefully, because they are at a lower risk of developing plasma leakage and therefore progress into that dangerous, being at risk of having more severe dengue. Now, what could these risk factors be? And we believe that they could fall into these 4 domains. Either they may be predictable clinically, or there may be virological risk factors that we can identify and to help identify patients that might progress to plasma leakage. Maybe it's something to do with host genetics, or maybe it's just immunology. And it may be a combination of these factors. So that is what we're actually exploring. Can we identify, before plasma leakage happens, in the early stage of the illness, risk factors in any of these domains that might help us to identify who will get plasma leakage, and therefore others could be, so at least to minimize the unnecessary admissions at times of epidemics. So to do this, you ideally need a cohort study where you recruit patients before plasma leakage happens, ideally in the first 3 days of fever. And then you take your samples and then you do and collect your data and follow up these patients during hospital admissions on a daily basis and record all the investigations and what happens to these patients. Did they get plasma leakage or didn't they get plasma leakage? And record the outcome. Was it severe dengue, just plasma leakage only, or undifferentiated dengue fever? So then, of course, you will have 2 groups who had plasma leakage and who didn't have plasma leakage. And then you can go back and see, were there any differentiating factors in the first 3 days of illness that could have differentiated the group with plasma leakage. And that is exactly what we are doing in our prospective cohort study called the Colombo Dengue study, in collaboration with the University of Colombo, Sri Lanka, and WISP. And we recruit patients from the National Hospital of Sri Lanka in Colombo. And this is a map of Sri Lanka. And you can see the Colombo district here. It's the smallest district, I think, in Sri Lanka. But this is the district with the highest population density. Both the administrative capital and the commercial capital of the country is located in this district. And the color code here shows the average dengue incidence per district between 2012 and 2016. The more intense the color gets, the higher the incidence. And you can see, even if you take that 5-year average, Colombo district had the highest dengue incidence in Sri Lanka. And that is where we are getting our patients from. And patients are recruited in the first 72 hours of fever. And the samples that we take are serum and Buffycoat. And they are followed up with their and to see who will develop plasma leakage and who. And this is not an interventional study. We don't intervene, and we let the physicians manage their patients, but we observe what happens. And then in subgroup of patients, we do a post-discharge interview at 2 months. So it's currently ongoing. So I'm going to present some of the interim data that we have analyzed. So it started in 2018. So far, we've recruited 877 patients. And of these, 523 were confirmed dengue patients. So we confirmed the disease by the NS1 antigen testing or RT-qPCR. We usually do both on all samples. And there were 140 patients with plasma leakage, and of those, 25 ended up having severe dengue, according to WHO 2009 criteria. And we also ended up with 354 non-dengue fever patients. They had an illness similar to dengue, but their dengue diagnostic tests were negative. And they remain undiagnosed to this date. And this group is also important for us because we have another project, hopefully, to discuss that in the last slide, because that's not the main focus of this talk. And just to remind, if patients were made an alternative diagnosis during their hospital admission, they're already excluded. They will be excluded, like leptospirosis, for example. So these 354 patients are undiagnosed patients. They didn't have an alternative diagnosis. They had an illness similar to dengue, but we don't know what they had. Now, the first stage of recruitment, as you can see, is now complete, and the second stage is now planned and is actually approved. And we hope to recruit up to 2,000 patients in this stage. So these are some of the projects that are currently going on, and I will explain these projects one by one, and probably I won't have time to explain the last one, but we'll see. Now, I mentioned in the first instance, we were looking for risk predictors in 4 domains, and the first domain was clinical risk predictors. And if we can actually predict who will get plasma leakage clinically, that would be the most useful thing, because remember, any tool that we find or risk factor that we find should be applicable in a low cost, low to middle income country setting, because those are the environments where dengue infection is quite rampant. So, and if we have expensive tools, even though they have risk predictive value, they may not be useful to the people that matter. So clinical risk prediction is ideal. And so that is something that we did with an interim analysis to see whether early clinical signs or symptoms or basic laboratory investigations can predict plasma leakage. And we compared patients with dengue with plasma leakage versus others, and etc. For the second comparison, we didn't have adequate power. As you could see, the severe dengue numbers were quite small. But to cut a long story short, there was only one predictor that we found. So that's the clinical risk prediction or using basic laboratory investigations doesn't tell you the whole story, or it's not as useful. I mean, just one risk predictor that we found. But nevertheless, this could be just part of the puzzle, and in a scoring system, this could be one element. And it's just that we have to look in the other domains also. So what we found was that a early and significant increase in AST levels. AST stands for aspartate aminotransferase. That's an enzyme found in the liver hepatocytes. And that's a basic liver function panel. That test is included. And so that was elevated in patients with plasma leakage significantly. And that was independent association. So AST may have 2 sources in a dengue patient, so it may come from the liver or may come from the muscle, but there was a non-significant increase in another enzyme found in the liver, ALT. So therefore, this is probably coming from the liver, and it may be possible that patients who get plasma leakage have an early hepatitis in dengue. Now, this is another side project that we did. So I'm not going to details of these 2 projects because it's already published and you can read about it, and there's the citation is given below. Now, one of the major problems in diagnosing dengue is the diagnosis test itself. So usually this NS1 antigen testing for the viral antigen NS1 is a bedside test, and it's a qualitative test. But it costs about $10 US, I think, the commercial price of that test. And when you have case loads in hundreds of thousands, the government cannot afford that, and in those on those settings, and certainly the public cannot afford it. I mean, majority of people may not be able to afford it either. So in that circumstances, NS1 test availability, even in Sri Lanka, it's a middle income country, varies from time to time. Sometimes government provides the test free of charge in the public healthcare service, and sometimes it's up to the patient to get it done. And then, of course, you can see the sensitivity and specificity varies depending on at which stage of the illness that you do it. Now, the other option is to do RT-qPCR. Now, that is definitely too expensive. So there's no way you can do RT-qPCR for diagnosis in that setting. So what we assessed here, now, by the way, I must acknowledge that this data was generated by a group of collaborators in association with CDS. In University of Calgary, Canada, and Stanford University, and we helped with the analysis. But this data was generated by those collaborators. So what they assessed was whether RT-LAMP, or reverse transcription and loop-mediated isothermal amplification, is a better alternative. It's not as simple as NS1, but it still can be done without sophisticated genomics laboratories. It needs a reverse transcription, but there's no need for a thermal cycler, because the genomic amplification happens at a constant temperature, and as more amplicons are made, pH changes in the solution, and it can be read as a colorimetric reaction. And if we assume RT-qPCR as the gold standard, and you can see RT-LAMP here has a better sensitivity and specificity compared to the NS1 test. And if we assume no gold standard, because RT-qPCR is far from a gold standard actually, then of course you can see sensitivity and specificity of RT-LAMP increases. And that also has been published, so I won't go into much detail, so I'll just leave it there. And I'll talk about the unpublished data from this point onwards of other projects that we are doing. So this data is still unpublished, so please don't share. Okay, so triaging for ultrasound scans. Now, what does this mean? Now, I told you critical phase is when plasma leakage happens. And the traditional marker to identify plasma leakage is 20% increase in hematocrit. But there's another alternative. You can do ultrasound scans just to visualize the fluid that has leaked. So you can see fluid in the pleural cavity or the peritoneal cavity, and that's direct visualization of fluid. And if the fluid is there, it's there. So you know it's plasma leakage. But there's a problem because there's a high demand for ultrasound scan in times of epidemics. For example, easily when I used to work in National Hospital of Sri Lanka, there may be like 160 patients per day. If we were to do ultrasound scans for everybody in 8 medical wards, male and female, so that will be 16 units. So easily you will get up about more than 150 patients for ultrasound scan. And the patients may need to be scanned more than once per day because we are talking in terms of hours. And the radiology department would be like overwhelmed if that is the case. And that is a 3,000 bed hospital. So there are other patients who are non-dengue patients who also needs to be scanned as well as outpatients. So that's not going to work. So now there are two questions here. Is ultrasound better than hematocrit in identifying patients with plasma leakage? And if that is better, how do we prioritize which patient gets the scan? So that is what we are trying to answer here. Now, is ultrasound scan better than hematocrit? Now, first of all, this is data from our cohort actually. So this is plasma leakage detected by ultrasound scan versus plasma leakage detected by hematocrit, and there is a strong association. So what that tells us is that if you are positive by the hematocrit criteria, you're very likely to be positive by ultrasound also. So there is a strong overlap there, so no surprise there. But this diagram here will show you the difference. Now, the 4 categories on the x-axis is hematocrit rise less than 10%, between 10 to 15% from baseline, 16 to 20%, and more than 20%. Based on the traditional criteria, only this category will qualify as having plasma leakage by hematocrit criteria. And the 2 colors here are patient with plasma leakage based on ultrasound and patients who didn't have plasma leakage on ultrasound. And you can see the hematocrit has a degree of sensitivity and specificity, therefore, so it's not a perfect measurement. And even when the hematocrit levels were less than 20%, there were patients who had plasma leakage by ultrasound. Now, we agree that ultrasound, so therefore ultrasound may be better, but how do we prioritize? And any prioritization system that we develop should be deployable in those settings in a low-cost manner. So we devised a derivation cohort as from a part of our dengue cohort. So we selected 535 patients with clinically suspected dengue, 308 with confirmed dengue infection, and of them 96 had plasma leakage detected by ultrasound scan. And then we look to see were there any clinical predictors or laboratory investigation predictors that was positively associated, statistically significantly associated with ultrasound scan detected plasma leakage, and we found 3 independent associations in the early days of fever within the first 3 days. A platelet count less than 100,000, AST level again, and then having abdominal pain. The weighting of these 3 associations are different, and we have devised a scoring system based on those weightings. And currently a validation cohort is in progress to see that scoring system, if that scoring system works. And another project that we are doing, so remember we are going to look for risk predictors in 4 domains. And I already mentioned that in the clinical domain, we only found 1 risk predictor. And the second domain was virological risk predictors. And to identify virological risk predictors, first of all, you need to do some groundwork, because we don't have a assay to amplify the dengue genome as a single whole amplicon. At least we didn't have one until we developed one. So this here is the traditional way that dengue sequence amplicons are made, and all those full genome dengue amplicons that you see in public databases were made this way. So there's the dengue RNA transcribed into the cDNA, and then it's amplified in 4 overlapping PCR reactions. Now this is fine if you are doing Sanger sequencing, because per sample you only get 1 sample and it doesn't matter whether you amplify them in fragments or not. But if you do next generation sequencing, it has this additional capacity to detect within host variants. And if your genome is fragmented, then it's very difficult to say which part goes with which variant. And therefore, a lot of analysis that could have been done with next generation sequencing is lost because of this fragmented approach. So I think in 2017 and 2018, we developed a full amplicon generating assay, avoiding this fragmented PCR approach. And that generates a complete single amplicon without fragmentation, starting from RNA to a double-stranded DNA conversion in a nested PCR. And this assay is now working well in our hands, and our PhD students, Tiruni and Braulio, are quite conversant with this, and it's working well. And a manuscript is currently submitted on this approach. Now, generating the amplicon is part of half the problem sorted. Now, the next problem comes with regard to sequencing. So I told you next generation sequencing offers some power, but the most popular next generation sequencing technology, Illumina, is actually a short read technology. So when we want to prepare libraries for Illumina sequencing, that again results in fragmentation of the genome. And so that advantage of that generating that whole genome is lost. So fortunately, we have this other 3rd generation sequencing methods now. The one we are using is something called ONT, or Oxford Nanopore technology. That is so those technologies are capable of generating full-length genomes, long reads, and sometimes to the extent of 100 kb. Now the dengue full-length genome size is about 10 kb, so ONT is perfectly capable of generating a single sequence in the full genome as a single read. Now it may be cheaper compared to which technology you are using in ONT, but the main advantage is portability. Now the smallest sequencer of this ONT technology is called MinION, and that's a handheld USB pluggable sequencer. And you don't need a very sophisticated genomics laboratory to sequence the virus based on this technology. So whatever virological risk predictors that we find in our labs need to be deployed in a low-cost setting in the countries that matter, and therefore we cannot rely on very expensive sequencing technology. Now, PacBio is another technology which can do this kind of long-read sequencing, but it is quite expensive and definitely out of reach of many of those countries. If we identify our risk factors using that technology. So whatever we want to do with a technology that is transferable to where it matters. Now, there is a caveat, however, there's a problem in using ONT. It has a relatively high error rate. And so the initial groundwork that we did was to benchmark ONT sequencing against Illumina sequencing to see under which circumstances we can accurately use ONT sequencing, minimizing the error rate. And we have a PhD student, Nazir, who did an entire PhD on how to reduce the error rate in ONT reads and to bioinformatically circumvent this error rate, and he has got some great results. And over the last 3 years, the company itself has improved their sequencing technology, so the error rate is now much better than what it used to be in 2017. So there's a lot of promise in this area. So these are the results of our ONT and Illumina comparative sequencing. And we had about an average of 1,000 read depth per sample, and we sampled 33 patient samples with sequenced 33 patient samples in both ONT and Illumina. And there is a 99.7% sequence similarity between the Illumina generated consensus sequence and the ONT generated consensus sequence. And whether ONT could be as expensive as Illumina or significantly cheaper to generate these sequences, depending on the barcoding technology that we use. Now, barcoding means it's very expensive to sequence one sample per run. So what we usually do is we mix actually samples from multiple patients into a single sequencing run. But at the end of the day, we need to differentiate which read came from which patient. So we add a non-DNA barcode to these sequences, and that the technology of adding the barcode could be either ligation barcoding or PCR barcoding. If you use PCR barcoding, the cost could be significantly cheaper. But if you use ligation barcoding, of course, the cost may be comparative to Illumina. Now, the next step was to see whether that 0.3% mattered, because there was still a sequence dissimilarity of 0.3%. If we were to think Illumina as the gold standard, then that 0.3% would have been due to the inherent error in ONT. And if in a virus like dengue, where there's not much inherent variation, so even that 0.3% might matter when it comes to phylogenetics. So we wanted, since we cannot sequence all the samples with both Illumina and ONT, if we are going ahead with ONT, we need to know whether ONT would be accurate for phylogenetics. So we made phylogenetic trees using the same sequences, just sequenced on 2 different platforms. So on top, you can see Illumina generated trees, and bottom, you can see ONT generated trees. And this 70% indicates what is known as bootstrap support, because a phylogenetic tree is just not dependent on one tree. We make multiple trees, and then we see patterns that we see in all those trees. So the 70% means if 2 sequences are clustered together, that sequence clustered in more than 70 trees out of 100, and therefore then only they will be represented as a cluster in the final tree. So that's roughly what bootstrapping means. But anyway, the bottom line is you see an additional cluster here that you don't see in the Illumina tree. And as we make this tree quite conservative by increasing the bootstraps threshold, even at a 90%, you still see this, but at 99%, it disappears and the trees become identical. So even that 0.3% might matter, but just that you need to make your trees quite conservative, and that may help to generate the Illumina matching tree. And this is quite useful as a guidance when we don't sequence with both these platforms and just go ahead with ONT. So another thing that we done, I'm not going to go into details, is to exploit the real power of long-read sequencing, that we can have the opportunity to resolve within-host variants. And our bioinformatics research associate, Preston Leung, in collaboration with Nazir, has designed a tool called NanoQ, which can differentiate both hepatitis C and dengue within-host variants from ONT reads. And this is a phylogenetic tree actually made from within-host variants. Now, the traditional phylogenetics only uses consensus sequences, but now we have the opportunity to look into within-host variants as well. Now, all this has laid the groundwork to explore virological risk factor predictors. And we are hoping now Braulio, PhD student, is in the process of generating full-length amplicons from all our samples. So all the samples that we collect from CDS and aliquot is shipped to UNSW for further investigation. So we are using those samples to generate virological sequence, and we are going to examine all these parameters to identify to see whether there are any differences between patients who had plasma leakage or not. And we also get Buffy-Coat, so we are going to look at host genomics, preferably with the candidate gene pathways, to see whether non-gene pathways associated with this illness whether there are SNPs, which can predispose to plasma leakage, or if we have adequate power with ongoing patient recruitment, we might do a GWAS. And immunological risk predictors is another domain that we haven't still explored, but we are going to see whether the cytokine profiles were different in patients who had plasma leakage or not. NS1 antigenemia, whether that plays a role. And recently a paper has come out to say that Zika infection is a risk factor for severe dengue. And we are going to look into that as well. And remember that I told you there were non-dengue patients with fever where they remained undiagnosed, and we are hoping to use those samples as a standardization tool to develop a low-cost metagenomics diagnostic pipeline for fever in the tropics. So those are future directions. And we have a few PhD students working on this, as well as few PhD students due to start on this work. And this is the space to watch out for in future. So I'd like to acknowledge a lot of people who have collaborated over the past 3 years on this project. So from this, Rovina, Andrew, Fabio, Preston, and all our PhD student, and Braulio, Tiruni, Nazir, and who did all the Illumina sequencing of dengue sequences. And on behalf of University of Colombo, the chief investigator of CDS, Professor Deepika Fernando, the clinical lead, Professor Senaka Rajapakshan, and special mention of the PhD student, Chaturaka Rodrigo, who's doing bulk of the work back in Sri Lanka in maintaining all these databases, interviewing patients and collecting specimens. And all the patients from the National Hospital of Sri Lanka and all the collaborators, and some of them were direct collaborators of us and others through the CDS investigators in Sri Lanka, and also the funding agencies. Thank you very much.
All right, thank you very much, Chaturaka. So I'd like to invite anyone to ask any questions, or you also have the option of liking some of the questions that have been asked. So we have a number of questions, Chaturaka, and I might just run through the most popular ones to start with. So I think as part of the lab investigations, did you look at infecting serotype and identify parity of infections, so primary, secondary?
Yeah, so infecting serotype, we have looked at it. We do RT-qPCR, you can actually serotype the infection. But there's a bias in there because most of our samples came from sort of like a hangover from that 2017 huge epidemic. And most of our samples are dengue serotype 2. So the other serotypes are quite few in number. So we could only analyze dengue serotype 2 versus others, and we didn't find any clinically significant association. Previous dengue infection versus the new dengue infections, we have not tested that yet, but we have stored our serum samples and we are going to do that analysis. But the interesting thing is we just don't want to know whether there's been a previous infection. We need to know what serotype that is. So we are just looking at a appropriate assay to not just give us whether I mean, we can simply do IgG test and see whether the dengue antibodies are there, but we just need to know which serotype also. So that is a currently work in progress.
And I guess one of the questions that relates to that is we potentially look for co-infection.
Yeah. So we do look for co-infection, and that's because in the RT-qPCR, we put all the 4 dengue probes together. And there are a few co-infected patients, not many though. We have seen dengue 1 with 3, dengue 2 with 3. But the numbers were not didn't have enough statistical power to see whether that contributed to disease severity or not. Very few co-infections.
Okay. And there are also quite a few questions, I think, relating more to the clinical presentation about the plasma leakage. So I think there was some asking what exactly causes the plasma leakage, and also could the albumin level be predictive of plasma leakage?
Right. So there are many theories as to what causes plasma leakage, but recently I think a paper has come out which gives some kind of significant evidence that it may be triggered by NS1 antigenemia in the patient. Now, of course, but that may be a bit different from the NS1 test that we do with the qualitative test, because we didn't see any significant association between having NS1 positivity with our qualitative test versus severe disease. But I think that test is just made for diagnostic purposes, and it's not really a pathological, not explorative investigation there. So that is something that we are going to look for using our serum samples. And in fact, one of our PhD students, Anurag, I think, had a very nice idea of how to test that, and hopefully we can go ahead with that. And I'm sorry, there was another part of that question, I think.
So could albumin level be predicted?
Yes, so the albumin level, so in the liver panel, we get the albumin level also. So the liver panel means AST, ALT, ALP, serum bilirubin, and albumin. And as far as I can remember, I can go back to my records. So that must have been analyzed, and probably that didn't turn up anything. So yeah, so I'm not sure whether it was done in all patients, because the full liver panel is not done unless it's specifically requested. So yeah, I need to go back and check on that. Yeah.
Okay. And I guess a question that sort of relates, I think, to the earlier one about the vascular leakage is, do you think there's any similarity between the vascular leakage in severe dengue and with sepsis?
Well, sepsis, of course, is a very complex phenomenon. So it's not, it could be, it's just not infection. So it could be like multiple other etiologies. So it's a very complex system. So as for dengue, well, the end result in terms of the end result, if it's not detected, and what could it end up with, like multi-organ dysfunction syndrome, and on the tail end, of course, the final common pathway is quite similar. But in terms of pathology, I'm not exactly sure, because sepsis is quite heterogeneous. And
there's a question here that relates to the patients, I think, that were undiagnosed. So I guess they had presented with fever, but weren't detected to have dengue. So is it possible that, I guess because they're both an RT-PCR based and an antigen test, that perhaps they just cleared the virus, and therefore that's why they weren't detected as having dengue?
True, that is a possibility, but remember, we are recruiting patients in the first 3 days of fever, and that is the time where you are likely to have viremia. So if you were recruiting at day 5 or day 6, of course, then that argument is quite true, and viremia would have disappeared. The problem, of course, is that patients may actually not know when may not remember when the fever actually started, because we are talking in terms of hours. So sometimes they may come in day 4 and say we are still in day 2 or something like that. So in that situation, yes, that may be a possibility. So before actually, if we are going ahead with that metagenomics pipeline and using all those samples, we'll anyway once we sort out our assay that we want to use, we'll definitely check for dengue IgM also in those patients, and then take the samples that are negative only for the metagenomics work.
Okay. All right, thanks, Chaturaka. I think that covers most of the questions that were asked. So I guess I'd just like to end today's webinar and thank Chaturaka very much for his presentation, and all the listeners as well for their questions. So thank you very much.
Thank you.